I wanted to point out, before John starts his talk, that the work he's going to be speaking about is it's actually NeuroPast, not NeuroFuture, but it's a really interesting program that was at the Allen Institute for about 3 years. It was a molecular genetics program, basically a molecular genetics looking at early developmental neural states of progenitor cells. And we don't continue this program beyond the 3-year time, so it's not something that you're actually going to see sort of in the future of the Allen Institute in terms of our website. But the work has been published, and it's really interesting, and I think a lot of the information that was derived from this as well continues to inform some of the work that we do, thinking about cell type diversity and cell type development in the cortex. So with that, I'd like to introduce John, and he's going to talk about marker discovery and modeling of human brain development.
Thanks, Amy. Hi, my name is John Mich. I'm a scientist at the Allen Institute for Brain Science, and the title of my talk is Marker Discovery and Modeling of Human Brain Development. Specifically, we want to understand the development of this beautiful and complex structure, the human brain, and how it arises from the progenitor cells, and also which aspects of its development are human-specific. So we've understood for a long time that mouse cortical development proceeds along a stereotype pattern from these immature progenitor cells in the ventricular zone. And during mid to late embryogenesis, you can see the inside-out deposition of layers resulting in the presence of most of the maturing cell types by around birth in the mouse. So this stereotype pattern is broadly conserved in the primate, but there's some important differences. First of all, the structures are much larger. There are many more cells present. And secondly, there's the presence of layers seen in the primate, for example, this outer subventricular zone here in orange, that contains primate-specific and greatly human-expanded specific progenitor cells, like the outer radial glial cells that have been very interesting lately, and which are thought to be uniquely important for the driving of human-specific brain development. So it's clear based on these comparisons that the mouse is just an insufficient genetic system to understand human-specific aspects of development. So in order to address this question, in our group, we undertook as a model system, we undertook neuralization of human embryonic stem cells to immature human brain cell types to understand their lineage. So that leads us to what was the Allen Institute Human Cell Types Project. The goals of our group were to build a genetically tractable system to understand the mechanisms of human cortical development and lineage, and secondarily to establish a robust and well-validated platform for the IPS and HES field. So this is the outline of what I'm going to tell you about today. First, I'll tell you about how we generate cortical neurons in vitro. Secondly, I'll tell you about how we did high-throughput gene expression profiling to identify the developing cell types in these cultures. Third, I'll tell you how we did in silico modeling to infer a conserved lineage of those cell types that are present. Fourth, I'll tell you how we experimentally tested those lineage predictions. And finally, I'll tell you how we compared our in vitro cell types to primary cell types to learn about their essential molecular character. So in our group, we had two distinct arms of human cortical neuron development that we had a system for. On top here, I'm showing you the excitatory neuron pathway that I'm going to focus on today. But also, in addition to that, we had a pathway for making ventrally derived inhibitory neurons that was just recently published. I'm not going to talk about that today. So in the excitatory platform that I'm going to focus on, first we start off from immature, undifferentiated human embryonic stem cells at day 0. And these undifferentiated cells are induced to form cortical neural cells by dual SMAD inhibition and inhibition of hedgehog signaling, so that by day 12, we have a nearly uniform pool of cortical progenitors. Those progenitors are then expanded greatly in response to these growth factors until day 26 to produce a mixed population of cortical and non-cortical progenitors. And then those progenitors are then differentiated in response to these neurotropic and neurogenic factors, produce a mixed bag of cortical and non-cortical excitatory neurons that we wanted to study. So we characterize these cultures by a number of different experimental means. One of which I'm showing here, which is bulk RNA-seq. So the data on this slide are from whole wells of differentiating human cortical lineages, all analyzed together by bulk RNA-seq. And that data is shown here in principal component space. And what you can see is that regardless of cell line of origin, H1 or H9 human embryonic stem cells, that the experimental time points cluster together and away from each other. Furthermore, the progression of the cultures goes through a stereotype path through principal component space, which suggests to us that our culture regimen really is a robust and reproducible technique. And using that data, which is a rich data source, we can look at the genes directly that are expressed within these cultures. We can see that housekeeping genes like beta-actin, turn on, are always present throughout the culture time course. Pluripotency genes like NANOG and OCT4 turn off rapidly with the onset of neuralization of these cultures. And at the same time, you can see the appearance of developing cortex and telencephalic genes like LHX2 and FOXG1, which suggests that we really are inducing cortical-like identity. But also at the same time, by about day 26 and maintained until day 54, we can see some unexpected genes turning on, markers of some ganglionic eminences like NKX 2.1, as well as some mid-hindbrain markers like INGRAIL2 and TFAP2. So it's clear from this data that there's a mixed regional identity of the cells that are being produced according to this protocol. So we need to take a fine-grained analysis using single-cell resolution in order to identify the cell types that are actually being produced in these protocols. So that's what I'll tell you about here. So in order to do targeted profiling of these these cells at the single cell level, we need a couple of molecular handles to be able to pull them up. So we made a number of different citrine knock-in lines into the actual human locus using targeted genome engineering. Two of those lines were the most useful to us. One, a SOX2 targeted allele, where we put citrine at the 3' end of the endogenous human gene to make a functional fusion protein of human SOX2, which made a useful reporter to target human neural progenitor identity. And also, we made a double cordon knock-in line with citrine fused to the 3' end C-terminal fusion protein of double cordon, so that we could prospectively identify maturing neurons in the human culture system. What you can see is that the SOX2 expressing cells start from nearly uniform appearance in the cultures and drop to about 50% of cells by day 54 of differentiation. At the same time, double cordon expressing maturing neurons are not present at the earliest time point, but become about 50% of the cells in the culture by day 54. So using these tools, we could undergo targeted sampling of cortical progenitors or maturing neurons at every time point during our culture assay. And so during the course of this work, our group performed hundreds of different differentiations of human embryonic stem cells to neurons. And those cultures were assessed by ICC and flow cytometry by routine criteria to assure their quality. One of the most important was the expression of EOMs, an intermediate progenitor cell marker, by day 26. And those cultures that looked good were dissociated single cells, sorted on the FACS ARIA into single cells per well in 96 well plates. And those single cells were subject to gene expression profiling by either cell-seq2 for our large main data set, or by a molecularly independent technique called SMART-seq2 for our smaller confirmatory data set. From those data sets, we then analyzed data in order to identify the cell types that were present within our cultures. So this is just a global view of all the cells that we identified by cell-seq. This is 2,684 single cells from all the time points between day 0 and day 54 in our cultures. And you can see that in principal component space, things look like they're working well. You can see the principal component 1 segregates cells according to progenitors on the left half of this plot, or postmitotic maturing neurons on the right half of the plot, with a few transitional cell types in the middle. You can see that the second principal component of the RNA expression among these cell types separates cells so that they stratify according to their times in culture. So it looks like our single-cell RNA-seq assay really does pull out biologically meaningful differences among cells. And then what we want to find is the actual cell types that are present in all the cells within all the time points. So here I'm showing you day 54 cells. We performed iterative weighted gene co-expression network analysis on those cells in order to identify discriminating genes that grouped according to discrete gene modules that describe those distinct cell types. And then those cell types that we initially identified were then subject to 100 rounds of random subsampling bootstrapping analysis to call final clusters of cells, which frequently co-cluster together within themselves and away from other cells. But you can see that some of the cell types that we observed, for example, this one and this one here, sometimes co-cluster together, whereas for example, this cell type here never co-clusters with any of these over there. So some of the cell types are more closely related to others. So this is, in total, we observed 41 distinct human early brain cell types at all time points of differentiation. So each column here is one of the cell types that we observed, and I'm showing you some of the distinctive marker genes that describe them. And these cell types that we observed during the cultures are named according to the time that we observed them, from day 12 to day 54. And they're also named with a P for progenitor, or an N for neuron, or T for transitional, or O for some of the other cells, some mesodermal cells that happen to occur to arise during the culture. And then also according to some of their distinctive marker genes that describe them. And the way that I'm displaying this here is that the cell types are grouped into closely related sets of cousins. In royal blue are these cell types that are neurons, or excuse me, that are progenitors of neurons that express high levels of these forebrain-like markers, such as LHX2. And in orange here are some distinct groups of cell types that express high levels of markers that suggest mid-hindbrain identity, like PAL3F2 or TFAP2D here. So it's clear that there are multiple distinct actual regions of human brain that are being formed in our cultures. So now we have this rich data set, and what we really want to do, the goal of the group is to infer concerted lineage. What is the developmental trajectory of all these cell types that we observe? So to recast it, what we want to go is go from this rich data set that took many person hours to generate, and infer a concerted lineage tree to understand the development of those cell types that we observe. So this is a difficult problem that's stymied the single-cell RNA-seq field for the last few years. There have been a few different lineage prediction algorithms that have been published in the literature. Things like that you might have heard of, like Monocle. But we felt that the extant lineage prediction algorithms had several important limitations that precluded their utility in our data set. Two of those limitations I'm going to tell you about here. First, those algorithms use distance metrics calculated with high-dimensional molecular data, all 20,000 genes. But we suspect that most of those 20,000 genes are actually irrelevant to the lineage development of most cell types. Like we know you can reprogram a cell with only a few different genes for its expression. And secondly, those lineage algorithms can only handle linear developmental trajectories, so just along a line. But we know that brain development is a branched lineal structure. So we've been working with Sharad Ramanathan at Harvard, who just recently published his new method for inferring lineage among cell types using single-cell RNA-seq data. And I'll give you a little bit of flavor of how his technique works. So his technique makes an assumption that there are only a few master-like genes that actually determine the lineage of cell types. And so we focus our analysis on transcription factors that are differentially expressed among the cell types that we observe. Secondly, Sharad's algorithm imputes a new construct, which he calls triplets of cell types. I'll explain to you a little bit how this works. So suppose that you have three cell types, cell type 1, 2, and 3. And suppose that cell type 1 and 2 express gene A, whereas cell type 1 and 3 express gene B. And from this pattern of differential expression, you can discern that cell types 2 and 3 are likely to be further away from each other than either is the cell type 1. So you can draw this topological arrangement of relationship among those cell types. And so when you have 41 distinct cell types, you can discover all the high confidence triplets, the topological relationships among them, and then assemble them together organically using a Bayesian probabilistic model of maximum likelihood in order to generate a branched lineage tree. So what that algorithm tells us about, this is our in silico predicted lineal relationship among immature human brain cell types. You can see that the algorithm spits out something that makes a lot of sense. It starts from immature progenitor cells that we observed at day 12, which then proceeds down 2 branches into the LHX2 branch of forebrain-like cell types, or the PAL3F2 branch of mid-hindbrain-like cell types. And you can see that the main axes are down with sequential generation of different progenitor cells. And then at all the time points, the neurons pop off from the progenitor cells. So we have a collection of high confidence lineal relationship among cell types that we observe in culture. But the great thing about the algorithm too is at the same time, because you use the expression of the genes to predict those lineal relationships, you also have a short master list of likely high confidence master regulators that are the things forcing the cells on those lineal branches. So many of the papers that have been published using similar techniques would stop there, but part and parcel of our group's effort was to actually go and experimentally test those predicted lineal relationships themselves. So I'm going to tell you about how we did that using one way. We actually used two independent clonal analysis techniques to identify relationships between progenitors and neurons that are formed in vitro. I'm going to tell you about one of those techniques for the purpose of time here. And this was a bar-coded viral fate labeling assay. And so to do this, we generated a retrovirus that has constitutive expression of the TD tomato reporter, and also a degenerate 10 base pair barcode in the 3' UTR of the retrovirus, so that the reporter enables sorting of single cells. We can process by RNA-seq, and then RNA-seq can also tell us the identity of the 10 base pair barcode within the cells, so that when you do single cell RNA-seq on these labeled cells, you can get their RNA content to identify their cell types, and from the barcodes, you can infer their clonal relationships. You can identify sister cells, sister neurons that arose from the same progenitor mother cell. So how we implemented this was that we infected with this barcoded virus at low MOI at day 27, just after the day 26 replating of progenitors, and then allowed the cultures to differentiate till day 54 when we sort and score single cells by RNA-seq. And here I'll just show you two pictures of what some of those clones look like by the TD tomato expression. You can see that they grow over time like you would expect, and that there are some distinct morphologies among those clones. This one primarily immature and possibly immature glial, and this one primarily neuronal. So we applied this technique to multiple different differentiations, sorted and scored hundreds of individual cells by RNA-seq. And from those hundreds of cells, we identified 41 unique clones that span multiple cell types that were present within our data set. And each clone here is along one row. And this plot shows you the appearance of which cell types were present within which clones. So when I'm showing it like this in a suggested way, grouping the PAL3F2 cell types together here and the LHX2 expressing cell types here, you can see that the PAL3F2 cell types very likely tend to co-occur together with each other, and the LHX2 expressing cell types also tend to co-occur with each other. And furthermore, they are far less likely to segregate, far less likely to get PAL3F2 cells along with LHX2 cells within the same clone than you would have expected by chance. However, there are a few, a small minority of clones that do have both LHX2 and PAL3F2 expressing types within them. But when you look at them, you can see that they're actually there are much larger clones. They contain many more cells within them than the unibranch containing clones. So we suspect those few bi-branch potential clones likely arose from a few immature progenitor cells that persisted till day 26. So I'd like to just summarize that we did two independent clonal analysis experiments that both each independently support the fact that the PAL3F2 LHX2 lineage branch is a real phenomenon and occurs by day 26 in these cultures. So I'd like to finish up finally and tell you about that we think that this is a real thing that happens in these cultures. And finally, we want to compare those in vitro cell types to the actual brain cell types in order to understand what do they represent in vivo. There are two questions we want to answer. What is the identity of our cells? Do they correspond to any known physiological cell types? And secondly, we want to be able to measure the molecular similarity of the in vitro and in vivo derived cells at the single cell level to understand how real they actually are. So I'll show you at the Allen Institute, we actually have an excellent pipeline to get to procure precious resources of human brain tissue. Here, post-mortem prenatal human brain tissue, which is a precious resource for these kinds of studies. So here I'm showing you these tissues stained with SOX2, a pan-progenitor marker. You can see that it stains forebrain ventricular zone cells as well as hindbrain ventricular zone cells. So it looks like a real pan-progenitor marker like we would expect from mouse studies. Whereas LHX2 expression is confined to only forebrain ventricular zone progenitors, but completely absent from hindbrain ventricular zone progenitors, suggesting that in humans, LHX2 really is a forebrain specific progenitor marker. Like these cells probably are real forebrain progenitor cells. Also, we want to look at markers of neurons. Here I'm showing you some cartoon representations of cortex versus hindbrain. And then when we look for markers of some of the maturing cell types, for example, NFIA expressing neurons that are present within the LHX2 branch, when you look at them and score where they're present within these two cultures, you can see that there's abundance of them throughout the prenatal maturing human cortex, but only relatively few of them throughout the prenatal maturing human hindbrain, suggesting that these NFI expressing neurons really are human prenatal cortical neurons. In contrast, when we stain with PFAT2B markers of some of these Pal302 branch neurons at day 54, we never observed any of them throughout the cortex. We could see an abundance of them throughout the hindbrain, suggesting that these really have molecular similarity to actual human hindbrain neurons. So it looks like the branches of our inferred lineage pathway really do reflect differences in regional patterning among human brain cell areas. So with that in mind, we wanted to then say, okay, within those dorsal forebrain cells, how similar are they to actual real live human forebrain cells? So we focused on the LHX2 branch of cells for this analysis because it's easier to get cortical neuron, single cortical neuron. And in order to do this, we utilized a technique that we have developed in our group called CRISPR, which is a technique for targeted profiling of single in vivo cells using antibodies. And we wanted to compare those cells that we could isolate from in vivo tissue to our in vitro cells that we generated RNA-seq data on. And so each of these rows in these heat maps is a single cell, and each of the columns is a single cell, either from in vivo tissue or from the in vitro cultures. Each row is a gene. And these genes are organized into modules. And what we can see is that there are a few modules of genes that are actually very well conserved between the in vivo cells and the in vitro cells. And those modules tend to group according to pan-progenitor markers like SOX2, intermediate progenitor cell markers like EOMs, or pan-excitatory neuron markers like double-corpus 25. So we think that our in vitro LHX2 expressing cells really do have high molecular similarity to the in vivo tissue-derived cells. But that's not the entirety of the story. We also observed a number of gene modules that are actually divergent between the in vivo and in vitro tissue. I can't go through all of this, but I want to point out a couple highlights. First of all, some of those things that we observe in vivo that we don't observe in vivo include these markers of neuronal maturation, like NeuroD6 and SAPI2, which are expressed at high levels in vivo, but very weakly in vitro. And even markers of maturing progenitor radioglial and outer radioglial cells, markers like GFAP, are present at high level in the in vivo progenitors, but only weakly expressed in vitro. So it's clear that the LHX2 forebrain cell types really do have molecular similarity at the single cell level to their in vivo counterparts, but they do have some deficiencies. They don't fully recapitulate the in vivo expression profiles. So with that, I'd like to summarize the results from the in vitro human cell types project about excitatory human neuron development in the in vitro system. We believe that the in vitro model really does recapitulate aspects of early human brain development. We see a major branch between LHX2 expressing forebrain cell types and Pal302 expressing mid-hindbrain excitatory cell types. And furthermore, we see that the in vitro LHX2 expressing single forebrain cells do show similarity to the in vivo counterparts, but they do lack some essential maturation markers. Finally, we generated a unified in silico lineage map for early human brain excitatory neurogenesis that we confirmed by 2 independent clonal analysis studies. And with that, I'd like to just summarize the talk with this cute art by Benjamin Facer at the Institute, where we show these adorable, fluffy little pluripotent cells coming down into the pluripotency station, and either getting on the orange brain train south, the mid-hindbrain parts of the brain, or getting on the royal blue train north of the forebrain parts of the brain. And finally, I'd like to thank all the people who contributed at high level to this work, whose names and smiling faces are shown here, as well as the Allen Institute founders, Paul Allen, Jody Allen, for the vision, encouragement, and support. Thank you.
Okay, that's great. So, any questions for John?
I'm wondering what's happening in this dish. Are these cells forming synapses on each other? Is there electrical activity that requires sort of differentiation?
Yeah, that's That's a great question. I actually omitted some of that data for the purpose of time, but we did perform both targeted patching and calcium observation of transients in these cells. The minority of cells are electrically active, but some are, and they also do have some molecular markers of maturing synapses, like Baskin and Homer. You can see closely opposed to each other among the cell types. But the calcium activity pathways were very immature, like you didn't see. There's high variability among cells in their profiles, and it's generally slower and less active than you would see in similar tissue from actual brain.
I wonder if you could comment on whether the barcoding method derived cell lineages have been were tested in a conventional in vivo arrangement, and whether those master regulators turn out to actually be so in vivo because you do show some cell types missing altogether, and then big chunks of the gene expression data look like lots of the genes are underexpressed relative to in vivo. So if the heat map just suggested that.
Yeah. So I think let me parse your question into two sub-questions. One is the gene expression profiles don't really match, and there's a lot of them. And it sounds like you might be suggesting that there are some master regulators that are present in vivo that are absent in vitro. It's very, very obvious, and it's very, very possible. This is a huge problem for the ESL modeling field. Is nobody has achieved fully mature neurons in vitro, and this is not just a problem for the modeling subset, but also an even bigger problem for the people who want to use these things clinically. And this is our work comes up against this wall, just like everybody else's. Every other paper has come up to this wall. So there's a huge lacking within the field about better techniques to make mature markers. And I think the first part of your question was about whether the barcoding lineage tracing can be applied in vivo to discern real in vivo transitions. We actually did try that a little bit using a in vivo tissue slice culture assay, but the clones that we recovered it could be done. We haven't done it, and we're not sure. It would have to be done in like an organotypic slice culture assay. But then once you take the tissue out and you put it in culture, then you're not really testing the same thing anyway. So this is one of the reasons why we didn't actually pursue that.
Okay, are there any more questions for John? We'll move on to Michelle.